Hello and welcome to my biology role model project. My name is Sophia Reynolds and I will be presenting on Hamilton Smith. Alright, so Hamilton, he was born in New York City, New York on the 23rd of August. And then 6 years later, his family moved to Illinois, where his father became a professor at the University of Illinois and his mother was a writer. So by those two, you can tell that they had very intellectual minds themselves, which they had passed on to Hamilton and his brother by introducing them to scholarly content at a young age, allowing them to play with chemistry set. As you can see at the bottom, the little Gilbert chemistry set that Hamilton has stated that he remembers playing with in the New York parks and in their home. And then along with that, his brother and him had a little lab in their home basement where they conducted science experiments. So you could say that this is really where Hamilton got that spark for science that he later continued into his career. And then if you look at his educational journey, he had attended high school at the University Laboratory High School of Urbana, Illinois, and he did so in 3 years. He graduated in 3 years, which he gives credit to his chemistry professors for, because he really had a special connection with them. And then once graduating, he went to the University of Illinois for a year to study mathematics, and then later transferred to the University of California at Berkeley in 1950 to study physiology. So he changed his major there. And then after that, he attended medical school at the Johns Hopkins University in 1952, and then received his MD from Johns Hopkins 4 years later in 1956. And then he went to work at the Henry Ford Hospital. Oh, my bad. After that, he went to work at the Barnes Hospital in St. Louis on a medical internship where he met his wife. And then after that, he went into the Navy on the doctor's draft for 2 years, and he spent his time in San Diego, California, where he said life was easy and relaxed, but quite boring. So he spent his time researching genetics, specifically population genetics. And then after his draft was over, he moved to Michigan with his family and began his medical training at the Henry Ford Hospital. All right, and then at the Henry Ford Hospital is where he found his preferred area of research, which was microbiology, and that consists to be the basic of his research still today. And then years later, he became a professor at the University of Johns Hopkins, where he still remains today, and he continues his research in restriction and modification of enzymology of genetic recombination, mechanism of bacterial transformation, and genetic regulation in prokaryotes and eukaryotes. Very sorry. And then in 1975 to 76, he went to study alongside another scientist, Max Bernstein, at the University of Zurich in Switzerland. And this, he said, was very inspirational to him, and he suggests that all scientists should go on their own and really get out of their comfort zone. All right. And then some career struggles that he had, even though he seems to be pretty consistent in his research, is that he actually had doubted his career beginning when he first was beginning college. As I stated earlier, he was originally going into mathematics, but he really struggled to find a connection with this and just didn't really have a sense of identity yet. And I really admire and relate to that, because as of right now, my major is definitely something that interests me, and I'm finding it to be quite accommodating to me, but you never know, I could always change my mind, and it's good to know that someone who is a Nobel laureate was actually never sure about his career either, and that's really cool to know. I also love how enthusiastic he is about his life, and about his research, and I want to be able to put myself into my work as much as he has. So, some of his hobbies include spending free time with his wife, his 4 sons, and his daughter. He also enjoys listening and practicing music, and he grew up playing the piano, so he tries to include that in his life a little bit, yeah. And then his most recent project that he worked on was published in January of 2022, titled Fundamental Behaviors Emerge from Simulations of a Living Minimal Cell. So they produced a whole cell, fully dynamical kinetic model of their cell JCVI Syn A. And so this cell was mainly synthesized to determine if it was really plausible to assume that the study of the simplest living cells will reveal principles that apply to all living systems. So their little cell had only 493 genes compared to the average cell that is composed of millions of genes, which is very impressive. And then they studied how their cell balances its processes such as metabolism, its genetic information, and its growth. So they ran simulations on a computer database called Brenda, as you can see in G on the in the picture to the right there. And then through these simulations, they collected they collected the fluxes in the metabolic reactions and the positioning of the macromolecules within the cell, which you can see in portion E of the diagram there. The little macromolecules are all the gray dots. All right, and then the novelty about this experiment is that these synthetic cells are smaller than any that have previously existed, and they hope that this becomes the new model organism instead of E. coli, like has been previously researched a ton. And then if you look over at the graph a little more, I can explain some of the parts. So if we start at A, this shows the ribosome locations and boundaries of their Syn3A cell. And then skip to C, you can see the membrane of the cell, which is shown in the green cubes, surrounds the ribosomes, the DNA, and the cytoplasmic space. Very sorry. And then D represents the addition of the array of membrane complexes and proteins. And then, like I said earlier, E shows all of the macromolecules in gray. And then if you skip to H, this is the medium that was used in their simulations to determine the uptake of nutrients during their simulations. And then I shows the GPU accelerant that the simulations needed, which predicts the number of active degradasomes breaking down mRNA, transcribing the RNAP, and then translating ribosomes. And then if you look to the graphs to show their data, you can see that N is the average number of times each gene is transcribed over the course of the 20-minute simulations. And then O is the distribution of the average number of times each mRNA type is translated in its lifetime. And then the result is that every mRNA is translated at least once in its lifetime on average. Alright, so something that they've uncovered so far during this research is that the synthetic cell, the DNA, is as usual folded around the ribosomes in a way that allows for the sequence order in the gene positions to be maintained. Their study has helped them understand how the cell balances the demand for metabolic processes as well as the genetic information and its growth over the cell cycle. This experiment ultimately helps researchers understand the principles of life for this minimal cell when little regulation is present, and hopefully it can become the new model organism used in research to come. All right, and then that was it. All right, thank you for listening. These are my sources. I hope you enjoyed, and I hope you learned something. Thank you for watching.